Okay, so although the title of my presentation is RNA-seq analysis to unravel the underlying mechanism of alder phytophthora interaction, I'm not going to talk much about this interaction, this specific interaction between these two organisms. Alder is a very important riparian tree species that is susceptible to the infection of several species from the Phytophthora genus. Our research, sorry, is focused on these two organisms, but I thought it would be more interesting to talk about the RNA-seq technique itself and its applications on forest diseases. So first, we have to talk about the importance of forest diseases. As you know, there are several kind of forests, several kind of ecosystems where we can find different types of important tree species, as we can see in the example, the chestnut, the alder, and the pine. These species are important due to the product that derives from this tree that is used by humans, for example, in the case of chestnut and pine, or because of their importance on ecology, that is the case of alder. These trees and many others are affected by different kinds of pathogens, commonly fungal or homicide pathogens, like in this case, Cryphonectia affects chestnut, alder is susceptible to Phytophthora alni species complex, and pine to Fusarium circinatum. So these diseases can affect the gel production of this product, or maybe affect the survival rate of the population in the habitat of these species, or many other factors. This disease becomes a problem when the speed of the spreading of this disease doesn't allow us to control, to manage these diseases. So it is so important to understand, to study the interaction between the pathogen and the plant, so that we can figure different techniques to manage this kind of interactions, to manage these diseases. So once the fungus is going inside the plant tissues or even inside the plant cell, there's a very complex interaction between the pathogen and the fungus. So here there's a very complex molecular dance where molecules from the pathogen and molecules from the plant play important roles. So there's a lot of molecules, a lot of genes that are expressing. When the pathogen enters the plant cell, it begins to express different genes related to pathogenicity, like genes to degrade the cell wall, or genes to degrade other compounds, to enter, to defense against the plant. The plant then detects it and begins to express different genes to defense against this infection. Also, the pathogen detects this detection and defense against this defense. So there's a lot of genes that they're expressing, and obviously we can notice that the transcriptome, the gene expression between a situation where the pathogen and the plant are not interacting are very different between the gene expression when they are interacting. So just to take in advice, as you may know, when a gene is expressing, it means that a gene is read from DNA, and there's transcription that is a synthesis of RNA from this gene. So here, if we have the RNA, the mRNA, this means that the gene is expressing. So here is where the RNA sequencing analysis enters. How does it work? Imagine that we have, for example, a pine that is, or an alder, or other tree species that is infected with a specific fungus. Another one that is not infected, it would be the control. So the process for do RNA-seq is, what we do first is to do an RNA extraction between, on both treatments, the infected and non-infected. Then we do a cDNA synthesis from this RNA to work with this DNA, but that comes from the RNA. And then we do a cDNA sequencing and quantification to know the amount of transcript. Now, remember that it comes from RNA, to know the amount of the different transcript that we have in each treatment. Then we do gene annotations, which means that we compare these reads with a database to know which genes we have in each treatment. So what we do then is a comparison between the transcripts in this situation and in this situation. What we will know is the amount of a specific transcript in one case and in another case. So we compare, like for example, here we expect that the genes that are responsible for the defense against the pathogen will be overexpressed. So the amount of genes here, the amount of a specific gene that is responsible for detection of the pathogen will be overexpressed compared with this case. So what we will do is simply like to know how many times are overexpressed these specific genes compared with another gene, or how many times are, imagine that a gene is inhibited in this situation, that could be also. So what we do here is to compare how many times this is inhibited compared with this. So what we have is what we call the DEX, the differentially expressed genes. The results we have are genes that are upregulated and genes that are downregulated. And also what we can have is a volcano plot that is a very common and easy way to see this kind of analysis, where we can see the upregulated genes and the downregulated genes. And also we can do some other analysis like the gene ontology analysis to know which group of genes are upregulated. For example, here are organized in the matter of the place of the cell where these kind of genes are expressed. So here we have a representation of cytoskeleton part, these kind of things. So what's the objective to do RNA-seq? We can do it for many things. Like, for example, one thing is to compare what I said, a plant that is infected and a plant that is not infected, or to study the differences between a resistant variety with another one that is not resistant, to know what genes are expressing the resistant and what the genes are not expressing the susceptible, or maybe to know the effects that can have viruses on the pathogen, because some viruses can have an effect on the gene expression of the fungus. So there's a lot of strategies that can be approached with this technique. But just to finish about this RNA-seq technique, there's an important thing, that is the RNA-seq is not just a research itself, it's a tool. So it's very important in RNA-seq to do further investigation. In the case I was telling you, like imagine that we are studying the genes that are responsible to the defense of the plant. Okay, we identified this gene that is very important, that is overexpressing, is upregulated in these 3 species that is resistant, or when we are comparing the control that is not infecting with this infected plant, what we can do is to, for example, select varieties that has more expression of this gene, or maybe it's not it's not very common because of the laws, but there are studies related to overexpress this gene with genetic modifications. So this is a very promising tool to understand and to study this interaction between the pathogen and the plant. So thank you so much for your attention.